Welcome back, guys. Last time we finished up, we talked about kind of an introduction to evolution, and we finished looking at Darwin's finches on the Galapagos. So today we're going to look at forces of evolutionary change. These items can cause evolution to occur in populations across species, and I think we'll be looking at five different varieties of forces that can force this type of change. So today we're looking at forces of evolutionary change. So forces of evolutionary change. So again, last time we talked about evolution. We had defined evolution as the change in allele frequencies in a population over time. So we're just talking about, you can think of these as frequencies or percentages in a population over time, right? So essentially we're talking about the gene pool. And later we'll talk about a way that you can actually measure this. So if we think about gene pool, so when you think about gene pool, it's often helpful to think of kind of this image. So here we have these hogs, right? And we have these alleles. And if you take all these alleles from these hogs and put them together in a collection like a hat, that would be the gene pool. And we could then calculate the percentage of alleles that are the big dominant B and the recessive lowercase b. And that would give us some allele frequencies. So all evolution is just a change in these allele frequencies over time. So if we had more capital B's at year 5 or year 10, then we would know those allele frequencies are changing and the population is then evolving. So there's 5 ways that evolution can occur, 5 different mechanisms or forces. And the first one we've talked a little bit about, it's natural selection. And natural selection is going to be the survival and reproduction based on the response to these selective pressures in an environment that essentially end up driving what we call adaptations. And we've seen this with the finches. And we'll look at some examples here of evolution by natural selection. We have 3 different modes of selection. So we're going to look at 3 modes of selection. So we're going to go ahead and write modes of selection. We're going to sketch these out for you, have you get a look at these. So let's say that, and we'll just make an XY plane here. Make a graph. And then we're going to have, let's say, number of birds over here on our y-axis. And then let's go beak size here on our x-axis. And let's say this is in millimeters. And then we have a beak size that can range anywhere from like 5 to 15 with an average size of about 10. And let's say that we've got a population on an island in the Galapagos that kind of has just a normal distribution, kind of a bell distribution, something like that, where the average is right around 10. And then let's say that, so let's say these finches, they have a mean beak size of 10. So again, this is our original population. And let's say there's a drought, right? And this drought only leaves behind seeds that are quite large and spiny, and you need a really large beak to make use of these seeds. So what we would likely see is that our selection curve would shift to the right. And birds with larger beaks will survive because they can eat the seeds, and then they'll pass those genes on to their offspring. And those they can't, they'll die off. So it's likely that one, the number of birds is quite a bit smaller, but the mean beak size may now be up here closer to 15, might be like something like 13. And our selection curve might look like this after that selection event, right? Where this is then the new population or the adjusted population after the selection event. So this type of selection is what we call directional. Directional selection, and it's selection for just one extreme. So let's look at a second example of a different selection curve with some different conditions. So here we'll go, put that aside, and I'm going to redraw my figure, my graph. This time we'll include a different selection pressure. So I'm going to have a similar type of curve to start. And we'll have, again, number of birds. We'll have beak size in terms of millimeters. We'll have 5, 15, again, mean of about 10. So we'll have kind of this normal distribution here. Again, this is my original population. And then let's say that for whatever reason, small seeds were eliminated from the island and large seeds were also eliminated from the island. So what I have remaining is just kind of this sweet spot, this seed that only a bird with a medium beak size can make use of. So likely what I would see is, perhaps a similar number here in this range, but at the extremes, my population is going to be severely limited. So my curve might resemble something like that, where everything is kind of focused around the mean, and this is my new population. And what we would call this stabilizing selection. So this is stabilizing selection, and it's selection for the average and against the extremes. Right. And if you think about human birth weight, it's a good example of stabilizing selection. When you look at developing nations, the human birth weight is going to be around 6 to 7 pounds. And if you get births that are way below 6 to 7, 3 and 4, you're going to have issues with lung development in premature births. So you're going to see a higher mortality risk there where in developing nations where you don't have great neonatal care. And then if you go up here to the 10 to 15 pound range, in developing nations, you're going to see a greater risk of the pregnancy to the mother and the child with complications. So again, stabilizing selection would have average birth weight right around 6 to 7 pounds. Let's look at a third type of selection. And again, we'll start by resketching the graph. So we got number of birds here. We got beak size. Again, 5, 15, 10. Let's say my beak size, again, is like the original. So there's kind of my standard distribution, my original population. And now we have seeds that are available on this island that are really small and seeds available on this island that are really large. And there's nothing kind of in the middle for birds at the mean beak size. So I might have a peak of, beak size here, and then I may have a peak for beak size here, and just a giant trough, a dip down here. So my curve might look something like this, where birds are going to thrive at these extremes and struggle at the mean. So again, this is my new population. And what we would call this is disruptive selection. This is a disruptive selection curve. And this is selection for both extremes and against the mean. Right? So that would be a third type of selection curve. And those three curves, again, are natural selection. You have selective pressures that are shaping those traits and pushing the curve to either be directional, stabilizing, or disruptive. The second mechanism by which evolution can occur is what we call genetic drift. And this is more of a random chance disappearance of alleles rather than a selection. So this is not selective. So it's not selecting based on criteria or alleles. And because of that, it's not adaptive. Individuals that survive are not going to be better suited to their environment. It's just kind of random chance that they ended up in a scenario where they're surviving and others aren't. So we'll look at a couple of different examples. So the first example is called founder's effect, and we can use the finches as a good example of this. So let's say that we've got finches here, and let's say this one's red, and then most of them on Ecuador, the coast of Ecuador here, most of them are like brown, and maybe one out of like 10,000 finches are red. But it just so happened that, let's say that 10 finches were pushed out to the Galapagos by like a major hurricane or something like that. And two of them just happened to be red. So two out of the 10, 20% that made the trip were red, when in reality, on the mainland, those allele frequencies would be significantly rare. Let's say 1 in 10,000, right? So if our real allele frequency is 1 in 10,000, and now over on the Galapagos, it's 1 in 5 because 2 out of the 10 were pushed out there, those allele frequencies on the island are going to differ significantly than the allele frequencies on mainland Ecuador. So that is what genetic drift can look like. Just because the population is small and isolated, that gene pool can differ from the main population, and that's essentially what evolution is, difference in allele frequencies. So let's go ahead and make a note of that. So again, our examples here are founder's effect, and we said that, again, a small population is where we're going to see this. That oftentimes is an isolated group. And we see this small gene pool it can differ from the main population allele frequencies. And that's what genetic drift can look like. There's going to be a second example here that we're going to look at, and it's called a bottleneck. And this is a way that, again, alleles can drift due to random chance. And it's called a bottleneck because it looks a lot like as if you're filtering the population through a bottle with a slim neck. So here we have a population where, let's say that we have in our initial population, maybe, I don't know, 30, 30, 40. So we have 30% pink, 30% green, or 30, 30, 20. No, 30, 30, 40. So probably need to change that a little bit. We'd have to go like, I don't know, 35, 35, 25 perhaps. So 35 green, 35% the pink, and then 25% red. That would be more realistic. So that's our initial allele frequency distribution here. And then as they're moving through this bottleneck, this event that's not selective but random, only the green and the pink emerge out the other side. And the red is completely gone then from the surviving population. And we just have really more, maybe it's 80-20 this time, pink to green, and the red is completely gone. That would be an example of a bottleneck. So let's look at a real example of this. So here's an actual example. So cheetahs experienced a bottleneck 10,000 years ago. There was an ice age. Many cheetahs died just because they did not survive this cold, and maybe only those at the very southern part of their range survived. And they also experienced another ice age 100 years ago due to poaching and habitat loss. Now, what this means is all the cheetahs, they share very, very, very similar allele frequencies and DNA. And it's as if they're all identical twins. You can take a skin graft from one and put it on another, and there's 0% chance that there will be a rejection issue with the immune systems of the recipient cheetah because they are all so similar. Now, this leads to some issues that we'll talk about here in just a little bit. Here's a second example, and this shows kind of some of the impacts. So here we have the greater prairie chicken, and you can see here in Kansas, we have 750,000 population size in 98, and we have almost 6 alleles per locus. So that's 6 different varieties of a trait or a gene per gene site. And we're looking at percentage of eggs hatched as a proxy of a function of what this allele per locus means. So almost 6 alleles per locus. We're getting 99% hatch rate. We go to Nebraska with a little bit smaller population size, maxing out at about 200,000. We have the same amount of diversity in the allele set, a 96% hatch rate. But when we go to Illinois and we look at the range reduction from 1820 to 1993, we're looking at a population size in the 30s and 60s, about 25,000, down to less than 50. And we're looking at the diversity lost going from 5.2 alleles per site down to just 3.7. And what does that mean? It means before we had 93% of eggs hatching in the 60s, and now less than 50% of these eggs are hatching in these Illinois-based prairie chickens, down from 99% in Kansas and 96% in Nebraska. So we have a reduction in the amount of chicks that hatch. So we have increased offspring mortality and decreased offspring survival. And we also have low disease resistance, and we see that in the cheetahs. So we'll note some of these things here, that again, a bottleneck is a large random reduction in population. And again, what we see, what does this result in? It results in low offspring survival and low disease resistance. Right, and we talked about 2 specific examples. We talked about really cheetahs, right, with the ice age, kind of 100,000 or 10,000 years ago, and then 100 years ago, right? And then we also talked about these prairie chickens of Illinois as examples of this bottlenecking type event. Number 3, we're going to look at, again, mechanism of evolutionary change is gene flow. So we'll look at an example of gene flow. So here we have 3 species of mosquito, right? And there's thousands of different species of mosquitoes in reality, but here we just have 3. We have kind of a highland mosquito up here. We have more of a river basin mosquito, and then we have kind of these plains-based mosquitoes. And the ones that live up at higher elevations, they have a resistance to a particular insecticide. So they have a gene that can metabolize or break down this active ingredient in this insecticide that the blue and the orange populations don't have. All right. And right now, if you were to treat these mosquitoes, the blue and the orange would be killed in large margins by this insecticide, and the ones up at higher elevations with the red population, they wouldn't really be impacted. So gene flow would be if we saw the red population start to breed with the blue and the orange, and we saw the increase of this resistance gene then as insecticides are continuing to be used, and those that don't have the resistance gene die off, and then the offspring of these hybrids that do have this resistance gene survive and increase in the population. So we can see this resistance gene going to increase. It's going to increase as that gene flows into the population from higher elevations. So a good example of gene flow here. So here's another example. Let's say that we have these deer, right? We have this mountain range that initially does not have a pass between it. So we have this eastern and western population, and let's say that they are able to interbreed. So they're separate species, but they're separated by this mountain range. And let's say that there is compatibility there in terms of breeding. They could even be a subspecies of one another. And then for whatever reason, an avalanche or a landslide and a pass becomes available. And now we have flow from western to eastern and vice versa. And we start to see allele frequencies start to change based on this hybridization and this gene flow that's occurring. So again, that would be another example of gene flow. So what we'll say is gene flow is the movement of genes or alleles into or out of a population. And examples here would be what we just discussed, kind of the mosquitoes and this resistance gene, and how we saw it increase once we introduced gene flow. Right, and then there was a little bit of a selection pressure then after that gene flow, but initially it would have just been gene flow, and then selection would have also started to take place. And these things don't happen in a vacuum. They're not completely isolated from one another. They can play off one another. 4 is mutation. Right, in previous units, we've discussed mutation in quite a bit of detail. So a mutation is just a change in DNA, which leads to a change in protein, which can lead to a change in the allele. And we've seen this right now with variants in SARS-CoV-2 or coronavirus. We've seen these variants emerge, and we're looking at evolution by mutation, right? Mutations are occurring. If the mutations are selected for, then natural selection can act on those variants, and those that evade the immune system are selected for and end up spreading, especially if they're able to spread with greater efficiency. So we're looking kind of at mutation in real time currently. Let's look at the last variety. The last variety is what we call non-random mating. If we take just a step back and talk about mutations again really quick. So here we have the original wild type Drosophila that has the sepia color eyes. They're round, and then a mutation to this can make the eyes bar-shaped and this cinnabar color. And then a second mutation can make the wings this vestigial shape where they no longer really function from the normal wing fly. Here'd be an example of non-random mating. So random mating would be if this beetle indiscriminately mates with all these others. Okay, it just is open to mating. It's not selecting based on any type of trait or characteristic. Non-random mating would be on the right. If there's preferential breeding based on shell color, and this guy only mates with this guy, or this female only mates with this male and does not breed with the others because of shell preference, that would start to swing allele frequency. So if this is the original set of alleles, and let's say it's 30, 30, or 33, 33, 33, right? 33% across the board. And then we have non-random mating or sexual selection occurring. We might see this shift to like 80, 10, and 10, or even more pronounced than that. And that would be a movement of allele frequencies by this non-random mating. So what does that result in? That results in something we call sexual dimorphism, where the male might have this big showy display and a female may be more drab and lack this showy display. Because males have to show females their gene quality, and they do this with some sort of physical display or this behavior. So that here we see these peacocks and this sexual dimorphism. And we can also look then at this distribution here, where males that have this huge tail, this big showy display, they have a really high reproductive fitness. But it may not mean that those individuals survive longer. It just means they reproduce way more often. Because if you have this big tail here, you might be more apt to be caught by predators because you're not able to camouflage as effectively. But they're going to have really robust genes that will communicate to females that they're good mates. So even at the sacrifice of individual survival, they will hedge that in return for a high fitness, reproductive fitness. Okay, so let's go back to our notes here. So we'll say that this is mate choice based on fitness markers. And we said that this could lead to something we call dimorphism, like we see with the peacock, or rituals or behaviors that are done to show dominance in terms of mating. So if you think about, like, you can look up elephant seals where they fight, and one elephant seal, one male might be the beach master, where he has rights to breed with all the females on the beach, and the other males do not have those rights at all. So he may breed 100 pups per year, and the others won't breed any. Or if you think about white-tailed deer and the dominance displayed in terms of the rutting behavior, or elk, that would all be because of non-random mating and the want to maximize your reproductive success. So that is mechanisms of evolutionary change. I hope that's helpful, and I'll see you next time.